All right, hello anatomy people, here to continue onwards with blood. So we've gotten to the point now where you should have reviewed your erythrocyte notes. We look at the idea of Lance Armstrong and the blood doping. I hope that video was interesting to you. I hope that made sense. All kinds of shortcuts and different strategies to reach certain athletic levels of performance without actually doing the work and putting in the training and the time. So again, I hope that was interesting to you. Now we're going to move onwards to the second type of formed elements. So we know that in normal blood composition includes plasma, the erythrocytes, and the white blood cells, and a little bit of platelets as well. But our focus today is going to be on the leukocytes. So leukocytes, leuko meaning white, cyte meaning cell, white cells. They're in the blood formed elements, so we call them white blood cells, right? Leukocytes are going to be much more diverse, much more complex than are the erythrocytes. So leukocytes are going to have an immune role. They're going to protect the body against microorganisms, toxins, and tumor cells. They remove dead cells and debris from the body. They're going to be complete cells. Right? This is a big contrast. Erythrocytes lost the nucleus. They lost the organelles as they matured into that, that complete erythrocyte. Leukocytes don't do that. Leukocytes retain their all their organelles, their nucleus, their Golgis, their mitochondria. So they're functional cells that can live long term. Some of your lab sort of activities, I'll try to incorporate this into homework as well. We'll deal with the identification of the white blood cells. If we were in an actual lab, I would have microscopes set up and you would kind of look at them, and I would quiz you on the different types of white blood cells. But we can accomplish the same thing with pictures, with drawings, and hopefully the homework will address that sufficiently as well. So we know that if we're looking at a normal smear, right, we have 55% plasma, 45% of the red blood cells, the erythrocytes, and then only 5% of the white cells. So they're not commonly found in circulation. They hang out in other parts of the body. We'll get to that a little bit later. But so they account less than 1% of the blood's volume. And for you numbers people, some ranges there. But the normal smear, very, very few white cells will be found in a healthy individual. So when we look at white cells, we just can't say white blood cells or leukocytes. We have to be more specific than that because there's actually 5 types of leukocytes. So we're going to hear about neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So again, these are the 5 categories of mature leukocytes, right? So of our of our formed elements, right? We have the 45% erythrocytes, very minimal amount of platelets, and then this sort of diversity of leukocytes in normal blood circulation. One of the first distinctions that we make is the idea of the granulocyte versus the agranulocyte. So we have one category, the leukocytes, broken up into 2 parts, the granulocytes and then the agranulocytes. So without agranulocytes, without granules. So cells without granules, granulocytes, cells with granules. So the granulocytes are the phils, neutrophils, eosinophils, basophils. The agranulocytes are the cytes, right? Lymphocytes and monocytes. There's a little acronym that's used often. It goes something, or mnemonic acronym, I don't know what you call that, but never let monkeys eat bananas. And never let monkeys eat bananas. I think they like bananas, but apparently you're never supposed to let monkeys eat banana, right? So that stands for never, neutrophils, let, lymphocytes, monkeys, monocytes, eat eosinophils, and bananas basophils. So that little mnemonic thing kind of helps you remember from the most abundant to the least abundant of these cells. So again, tells you the percentage. So neutrophils never let monkeys eat bananas. And again, it's just broken up into both granulocytes, agranulocytes, but it is useful to remember that. And is that question follows why? Why are they not all the same amount? Well, form fits function. And because they're in different amounts, it should give us an idea that they're going to have different roles, different jobs that they play in the body, right? So let me kind of get into the idea of the granules, or the granules, the granulocytes. So I'm going to come back to that here. I don't have a good picture here. Well, let me back up here. So when we look at the idea of the granules, so granules, if we go back to 1306, granules are basically lysosomes, or very, very similar, perioxysomes, little defense sort of organelles. They release different chemicals to help destroy things that are not supposed to be in the cell. So if that cell contains that organelle, the lysosomes, we say that they have a presence of the granules. If they don't have the lysosomes, they have the absence of the granules, agranulocytes. And the granules basically can pick up different types of stains. You can think of it like food coloring, different types of stains, acidic stains, basic stains, neutral stains. So that's how they would be stained and identified in a blood smear, depending on what stain that white blood cell absorbs. So it's going to be visualized by different colors here. So there's a visual color sort of distinction. There's an actual distinction. Granules are going to appear like little spots or like little chocolate chips, you know, in the cell. And then the shape of a nucleus. In 1306, they taught us that we have a round cell and we have a little round nucleus in that cell. But it's actually more complicated than that. And leukocytes are going to give us a, sort of an idea on that complexity here. So moving forward again. The development is more complicated again, but it starts with that same sort of generic cell. We learned about the hemocytoblast. We talked about red blood cells. Hemocytoblasts are the same precursor for the leukocytes now. It's a different pathway. The most direct pathway would be the hemocytoblast going to the lymphocyte path here. Yeah. It can go this way in a different series of pathways, you know, governed by different genetic signals, hormones, enzymatic reactions, inhibitors, a lot of different components we're not going to get into now, will then trigger the different types of leukocytes here, right? We have that one that's a little bit different, that agranulocyte, the monocyte, and then we have the three granulocytes, the eosinophils, basophils, and neutrophils. And eosinophils, to be a phil, P-H-I-L, means that you like or you love. So think of like we put a red stain, that the granules in the eosinophils pick up the red stain that we call eosin. So they love red stain. So they appear red. The basophils pick up the basic stains, so they appear blue, and the neutrophils don't pick up stain, acidic or alkaline. They pick up the neutral stain. So again, different colors. See the little granules and no granules over here. Right? So just, we're going to get better images here, but I'm going to cover this in a bit. But you can see once we reach the end to these granulocytes, that's it, they're done. The agranulocytes can continue development to a different type of cell. So lymphocytes can continue to form plasma cells. Monocytes can continue to form macrophages, and I'll address those shortly. But again, just showing you the the developmental history of these 5 types of cells here. So let's begin with the neutrophils. These are the leukocytes, or we call granulocytes. They're going to be the most numerous. And again, why do we suspect they're the most numerous? Right? Well, if we look down here, this gives us a big idea. So neutrophils are really, really good at identifying and destroying bacteria. And you can imagine, of all the things that we come into contact with, bacteria is probably the most common thing that our body has to fight off. So again, we have our bacterial specialists, hence then the most abundant type of granulocyte. They're also the first to show up during acute infections. So the first responders once we start to have these infections. They also are good at fighting off fungi, but again, we don't really come into contact as much, as often as, in absolute numbers with fungal infections as we do with bacterial infections. So again, the bacterial specialists, neutrophils, also can fight off fungi and basically different complexes that develop in the body that we'll get into in the immune system chapter later. So neutrophils. Neutrophils get a particular title. They're termed as having or being polymorphonuclear, PMNs. Poly mini, morpho shape, nuclear nucleus. So if you look at this picture here, this is a neutrophil. It's got little specks, little chocolate chip specks there. And then it's got this weird lobed nucleus. And that's what we mean by polymorphonuclear. It's not just a round circle. It's got these like lobes, right? It looks like little sausages kind of tied together sometimes. But so again, 1 lobe, I see 2, maybe 3, maybe 4, maybe 5 lobes there. And that's typical. They're going to have between 3 to 6 nuclear lobes. And if you were to ask me, well, why do they have nuclear lobes? That one I won't be able to answer you. I don't know how or why that helps the cell. Why does it have to be shaped in that weird way to function? I'm honestly not sure. I've never found any information. Maybe extra credit if some of y'all can go in there, find some research and let me know why does this cell function better with a weird shaped nucleus instead of a normal shaped nucleus, right? Yeah, bonus points there, right? So neutrophils, when they're called to action, think of like an ambulance. Think of the first responders. They typically hang out in their area, the firehouse over there, wherever. When the emergency happens, they're come out for a little while, do their job, and then when they're done, they go back to where they were. So these neutrophils do kind of the same thing. When they're called, they come out, they spend about 10 to 12 hours in circulation, and then they leave circulation and go into the tissues where the infection was, where the injury was. They're gonna do their job in that location and not in circulation. So the neutrophils. Eosinophils, right? So eosinophils are, again, one of the least common, right? Eosinophils are specialists against parasites, parasitic worms, tapeworms, hookworms, pinworms, flukes. So I don't know about you, but I'm going to guess that right now in a civilized society we have, very few people are infected with parasitic worms. I mean, in third world countries it's more common, but in our natural, clean environment, high hygiene environment, we don't really typically see a lot of parasitic diseases. Henceforth, there's not a lot of eosinophils ever in circulation. Right? So eosinophils, again, defend the body against parasitic diseases. So they're not looking for little microscopic bacteria. They're looking for these large multicellular worms that are in the body. They're also going to be involved in these allergic reactions, but in a good way, right? They're going to help reduce or control. They're going to help minimize, reduce the inflammatory responses that happen. So we're going to get into allergies a little bit later, but just no, this is a beneficial thing. This is a good thing. They help reduce the inflammatory response. So they're also polymorphonuclear, but they have a bilobed nucleus, right? And again, I use the color red, because they have red granules there. So again, red associated with this eosinophil. And I don't know about you, but every time I see eosinophils, especially when they're in the right position, they remind me of like a little alien head. There's a little alien head with, you know, the cool alien with these, you know, bilobe sunglasses there. Another student of mine a few semesters ago, his mind was in a different place, I guess, but he said that reminded him of a bikini top, right? So yeah, well, whatever works for you, right? So the bikini top, eosinophil, or the alien head with sunglasses there, right? This one over here on this side is the same thing, but the cell is shifted, so you're not seeing the lobes directly. You're seeing them from the side, from the lateral view there. So the eosinophils. Basophils. So basophils are the least common, and that's a good thing, right? Basophils are the alarm-sounding, dramatic little leukocytes that produce histamine. So histamine is notorious for producing allergies. So histamine is both an allergic sort of instigator, and it enhances the inflammatory response, which is going to be painful, uncomfortable. If you have allergies, your runny nose, you just feel stuffed, and it is all of that is caused by the histamine. So if you take allergy medication, a lot of the allergy medication are antihistamines, right? So, but when basophils feel the need, they remind me of little chihuahuas, right? You just hear like somebody knocks at the door, and or they don't even knock maybe, but the little dog just goes all crazy. That reminds me of the basophils, right? So they are small cells, these blue colored cells, and basically anytime they feel threatened, they release all this histamine and just cause a domino effect of chaos. So these basophils are counter to the eosinophils. So eosinophils kind of keep the basophils in check, and I should say the histamine in check. So basophils. So those are our 3 granulocytes. Now we progress to the 2 agranulocytes. So these look nice and clean, right? No granules compared to the granulocytes, the neutrophils, little granules, eosinophils, little granules that red, basophils, granules, blue or purple. So these are the polymorphonuclear. And then we have basically a regular nucleus to slightly modified nucleus here for the monocytes and the lymphocytes. So lymphocytes are the smallest of the white blood cells, physical size. They're the little, you know, the smallest, the little small stature compared to the other cells, right? They kind of crack me up a little bit, because you have a cell, a small cell, taken up by basically a huge nucleus, a nice round normal nucleus, right? It's like its brain is the whole size of its body, right? So it's got a lot of genetic information, and you can think of these cells as intelligent, right? And they have to be for what they do. So a large nucleus in a little cell is basically these lymphocytes. So when we look at the lymphocytes now, we're looking at 2 types, and I'm going to come back to these later. But for now, let's just say that we have the B lymphocytes and T lymphocytes. And what's the difference is where they originate from. If you were born in America, you're an American. If you were born in Mexico, you're Mexican, whatever. Yeah. So if you were produced in the bone marrow, you are considered a B cell, a B lymphocyte. If you were developed in the thymus, then you're going to be considered a T cell. So they're still lymphocytes. They're structurally similar, but they're going to have different training and different functionality later. Right? So B cells will eventually produce the antibodies. T cells are going to fight the body or help the body fight against viral infected cells and cancer cells. Now, this takes intelligence. You got to be smart, right? If I'm going to fight a bacteria, I know the bacteria is foreign. If I'm going to fight a fungus, I know that fungus is foreign. If I'm going to fight a worm, I know that worm's not supposed to be there, something foreign. But if I'm going to try to fight a cell, my cell that's infected with the virus, well, the virus is inside of the cell. So I'm not going to see the virus. I'm just going to see my cell acting kind of strange. And that lymphocyte has to know, man, that cell belongs to my body, but that cell looks like it's acting weird. It's infected somehow. So it needs to be able to identify normal, healthy, functional cells and cells that are from the body, but that are infected with something foreign. So again, takes some intelligence, and that takes that extra training that happens in the thymus. And cancer cells as well. Cancer cells are basically our cells. There's not something that we got infected with. It's our own cells that are just changing, that are acting differently, right? Different signals, lack of cell cycle regulation, that kind of stuff. So the T lymphocytes then must be able to distinguish this normal human cell, I should say not normal, but this human cell that belongs in the body is acting strange. So I'm going to go ahead and go and destroy it. So, and you can see if this is not regulated properly, the T lymphocytes could then start to destroy cells that are not supposed to be destroyed. So again, the big moral of the story, why do they have such a big nucleus for a little cell? Because there are a lot of brain, a lot of thinking and strategizing and things like that go on in the lymphocytes. The monocytes are kind of the big bodyguards, right? So lymphocytes are the smart ones. The monocytes are just the big, muscular, bouncer, strong type of fighters. That big U-shape kind of nucleus, the largest of the white blood cells. So they're physically large. And they can transform into something even larger and stronger, right? So monocytes typically are recruited a little bit later. So let's say it is a bacterial infection. So neutrophils get there to the site, they try to do their job for a few hours. If they're not successful, well, we got to call in, the backup. We call in the big, strong monocytes to come in. And to make them even more effective, think of a big, strong warrior now starts injecting with all these steroids to get even bigger, right? So the monocyte starts to modify itself and transforms into these macrophages, which are more stronger, more phagocytically able type of cells, right? So it's that secondary progression from monocyte to macrophage. And again, just big, very, very effective cells at phagocytosis. So I hope that makes sense, right? Our different types of white blood cells. It's not as simple as the erythrocytes, right? And I know this little video is getting a little bit long, so I want to, shut this one off soon. But I don't want to push this to the next group. So let me continue with the disorders here. If for whatever reason there is an abnormally low amount of white blood cells, we say that person is suffering from leukopenia, leukocyte deficiency, right? So there is a abnormally low white blood cell count. How can that happen? Well, certain drugs, maybe chemotherapy, maybe damage to the bone marrow, damage to the thymus. This can cause the white blood cell count to drop. On the, sort of the related idea of we have deficiency, well, what's the opposite of deficiency? We'll talk about leukemia. So leukemia is the opposite, it's a type of cancer. So the white blood cells themselves become cancerous, right? And they start to become problematic. So there's a bunch of them, but they're not correct. They're not functioning right. And they're not able to help the body fight off disease, bacteria, fungal, worms, whatever viruses, cancer. So basically, it diminishes the effectiveness of the immune system, even though there's an abundance of them. Now, there's about 70 types of leukemias. There's a bunch, right? And some are worse, some are, I'm not gonna say better, but they're not as severe. So we're gonna name them based on the type of cell that has become abnormal, right? So monocytic leukemia, lymphocytic leukemia. So if I back to this sort of developmental sort of process, if let's say down here, let's say we're looking at the eosinophil. What if this band cell right here, if this cell becomes cancerous and everything else is okay, well, only the eosinophils will be damaged and affected, but everything else is still able to work fine, right? So that, yes, it's cancer is not good, but it's not as severe as something else. Well, what if, let's say, this lymphoid cell became cancerous? So then that would have an impact on all of the lymphocyte lineage. So none of these would be formed properly. If, let's say, up here, what if this myeloid stem cell became cancerous? I hope you see then all of these 4 types of leukocytes would be affected in a bad way. If the hemocytoblast becomes cancerous, all of the white blood cells would be abnormal and the red blood cells would be abnormal as well. So depending on what level in the developmental pattern here is affected will depend on the severity of the leukemia. So again, so that's our condition. So leukemia is fever, weight loss, bone pain, can be accompanied by the anemias, because now you have too much white blood cells in the blood and not enough red cells, right? Can have bleeding problems because white cells now take up space that the platelets should be in there. So again, it just disrupts all the whole system, right? But with that, I think this is a good stopping point for this little video. So I'll stop it now, get it shifted, get it transferred, and then I'll record the platelets. And what do we have next? We also have the blood typing, and that's a really fun lecture as well. So we'll see if I can get to those today.